Okay. So what I have planned is to just talk through some key things from chapters 1 and 2 by pulling up the content objectives and having anybody who's present throw out questions that were conceptually challenging, like after you've taken the exam, what are you still struggling with? And then you can't access the exam questions anymore. You could see what you got wrong immediately after the first or second attempt. And I have to keep them protected because those questions banks were built in the spring term, and I can modify them, but I can't leave them open because they're shared, and so they have to be protected. But I can walk you through, 10 different questions that are in this bank, or so, however many we have time for, and
Yeah.
talk about how to answer them. So if you have a particular question that you remember struggling with, and I'm going to apologize. Normally I write my own test entirely, and actually by the time you get to exam 3, I said forget it, and I wrote the whole test instead of waiting for the group and then shared it with them, so they got my test and got to modify it. But in this shared situation online, some of the questions I've kicked out, after the fact. And so there will be a curve that I implement. But when I looked at your scores, I felt like this time around, things felt a little rough, and that I would just open it up for a third time and we'd do this review, and then I'd curve after the third. And what I will do when I open it up for the third is I will just tell it to take your high score. So there won't be risk involved in taking it the third time, if that makes sense. If you do worse, your score won't go down. And in fact, hopefully between the review, taking an hour after this to look through some things and then retaking it and seeing it a third time, and for a few people, it won't be quite the third time because they didn't take it the full 2 times, then you'll do a lot better, and you won't dump a ton of time into this, but a couple of hours, and then feel like you really are on top of this information, so that when the content resurfaces in different question formats on the final, you'll be prepared to like ace that section. Okay, so I'm gonna make this bigger right here on my screen now. If I zoom in a little bit, you'll see what we call our weekly content objectives. And normally they're based on a unit, but for online purposes, since we're doing like 8 or 9 weeks, we split them out into weeks. And so week 1 had chapter 1 and then the first part of chapter 2. And for most of you, you probably thought, chemistry, that's a breeze. And then you realized that this class is trying to stuff a chemistry review into like 10 days of learning. And I'm sorry that that happens. I promise I didn't plan it that way, and nobody does. It's how the course was designed. It's just a really intense chemistry review to get to relay some foundational knowledge, so that when you move on to chapter 3 in biological molecules, you have molecular structure and function in place. So when I look at chapter 1, the big chunks are the scientific method and how to understand the different components, what those terms mean, and how they are applied in real scientific scenarios. And that includes terms like independent and dependent variable, hypothesis, theory. Then we're talking about prediction, hypothesis testing, inductive and deductive reasoning. So it's all that terminology just rolled into applying to experiments. And so some of the stuff on the test is information based. Do you see something that looks familiar, and can you recognize a correct answer? But a lot of it is application of that knowledge to a new situation. And so you have a new word problem scenario, and 3 of the answers are wrong and 1 of them is right, and you've got to figure out what the best possible answer is. So multiple choice is nice in that the correct answer is always there, but horrid in that it's embedded in a sea of falseness or correctness, and you have to find the one correct choice or the one wrong choice amongst everything else. And so that's that's a pitfall of multiple choice. And then the second part is really looking at biology from an organizational and relationship perspective. So looking at the properties of life, or the 7 characteristics of living things, and then looking at prokaryote, eukaryote, and then the organization among living things, you know, from a single cell to, you know, what is the most inclusive unit of organization in biology, and that's where we're looking at the 3 domains. And so that's chapter 1, just kind of an overview in a nutshell. Within chapter 1, does anybody want to speak up and tell me what on the test conceptually was their biggest hurdle that they want me to touch on briefly?
I didn't really think I had anything to worry about. When I went into it, I was surprised I got such a low score because I understood everything.
Okay, so things made sense, and then in the application, it got a bit chaotic and confusing.
That must be it, yeah.
Okay, well, some of it is just in biology, the layers of content and information and terminology is way deeper than reading anything in a history textbook or a math textbook. And so then you have to take all that new information and use it in a new scenario. And that, if you haven't done that in a while, or, you know, you haven't taken a science class before or since, you know, high school, and if it's been a while, or if a lot of it was just memorization, then this college class is a new level. But it'll really get you prepared for anatomy and physiology so that you can do well. So did anybody have anything in particular they found challenging conceptually?
I don't remember the specifics.
Okay, so maybe when we scroll through a few things, you'll see that.
Yeah, I'll let you know then.
Okay, that works for me. Okay, so chapter 2 has, I've got it open right now to the first part from week 1, so atoms and molecules. So really looking at atomic structure, protons, electrons, neutrons, knowing that the electrons are in the shells around the nucleus, and then understanding how the behavior of the outermost or valence electrons affects the way an atom will behave, and whether or not it will form a covalent bond or an ionic bond. If it's a covalent bond, whether it will be nonpolar or polar, and then getting into electronegativity values. And beyond that, the next study guide or learning objectives were about the properties of water and pH. And so that would be on the next screen. And I see in the chat, there's a question about, there's a mention about the hypothesis questions. So maybe what we can do is pull up a few of those specifically. Thank you for mentioning that. Okay. So if I pull open week 2, and you're seeing what it looks like from my end with all my little green check bubbles. Okay, so yeah, it's water and pH, and then looking at biological molecules. So the 4 categories being carbohydrate, protein, lipids, and nucleic acids. And I'm sorry, I appear to have a little bit of recording feedback here. I'm not sure why, since I have my headphones on. Does anybody have conceptually something that stood out for them that was particularly challenging, like pH or the properties of water, or something about the biological molecules and structure and function?
Nope, Like I
Nope.
said, I understood it so well. I was surprised I got such a bad grade. I don't know
Okay.
what went wrong.
All right. So one of the things I've noticed, and I can't pinpoint, who exactly. When I read and I look at the study guides, which is a new thing that I started last term, and I had one for like every section, and then I decided I would just do like 3 and then give you a couple of study skills assignments, is I see some students really going in deep and looking at these content objectives, and I see some students just kind of touching the surface of it. And depending on which category you find yourself in and how you're using these content objectives, whether you're just glancing at them and going, aha, they make sense, or whether you're diagramming and asking yourself like deeper questions rather than just what is such and such and having to come up with the definition of the term that applies, that might affect your recall and how much synthesis of the information that you have. But let's just go ahead and open the test and spend, a half an hour looking at some questions and review. That may be just the best thing for you. All right, when I close that, do you still see my screen with the other pop-up? That's the test now?
Yes.
Week 3 cell structure, something.
Yeah, that's just, it's labeled for when it shows up. So I can, you probably noticed that the questions that you got the first time were not exactly the same as the questions you got the second time.
I figured that was by design.
Yeah, because we had to make a big pile of them so that, I don't have a lot of students who I've been particularly worried about cheating, but other professors have had that problem in the past. So they wanted to have a big question pool and then have questions be drawn from that. Last term, somebody somehow managed to get these enzymes questions into the second exam like 7 times over. And I was so glad that I caught it. It was like this funny duplicating glitch when you copy from one Canvas course to the another. And it was kind of sad and hilarious at the same time. And so that's all gone. None of that is in there for you. All right, so let's see. We'll just scroll through and look at some questions. So this one, which level of taxonomic classification will have the most types of organisms included within it? This is just getting at when you're looking at organization, which of these is the most inclusive? Kingdom, yeah. And so you have just enough time on the test that if you can't remember, if you have notes there, or if you have a PowerPoint open, or if you have the textbook open and you know where things are and you have a pretty good idea, you can take 10 seconds to check something, right, before you actually have to answer the question. It's supposed to be essentially closed book, but we're not doing any browser lockdown and we couldn't control what was next to you anyway. So for me, if you're learning while you're doing the test to find out what the right answer is, I don't care what you're looking up. Just don't share answers with each other, if that makes any sense.
It does.
Okay, so the next one. All scientific claims are supported or rejected by scientists solely on the basis of evidence. It should be true. Whether or not all scientists do this, well, that's debatable, but everything should be based on evidence-based. You should be able to provide support for a hypothesis or prove it false. And you can't just say that it's true because you think it's true. There's a lot of pseudoscience in the world, and even more with the coronavirus than ever before. Everybody thinks they're applying the science, right?
Oh yeah. Everybody's an expert.
Everybody's an armchair doctor and an armchair scientist, and
especially with all this time off.
Yeah, I have read enough of the primary literature in the last few months that has come out to know that we just have a couple of pieces of that 1,000-piece jigsaw puzzle right now. And things are shifting. My youngest brother and his wife have just recovered from the coronavirus, and they have no idea how they got it. Like, was it
You
the
never
grocery
know.
store? They live in
That one
Utah. They
bag
went,
they got,
well,
that one bag from the grocery store, that one sick person happened to touch before them.
yeah, but a lot of it is aerosol. We're learning, but I'm kind of digressing, so I should refocus and we could talk about the coronavirus.
I mean, we do coronavirus topic assignments.
We do, but let's do that like after the test.
Agreed.
Review. All right, so this one is about levels of organization, and we're talking about a Pacific Northwest rainforest. And so we've got plants, we've got insects, we've got animals, and it's talking about, you know, converting carbon dioxide in the air to sugar. So all of this stuff together, all these living things, what would you say this represents? So
I might have got this one wrong.
a population is a single group of organisms.
That one I knew.
A community is multiple groups, and a species is essentially all the organisms, all the populations of a given organism that might or might not be living together. So, you know, there are populations of gray squirrels, you know, that live here, that live in Seattle, et cetera, and they're all still part of the same species. But in my backyard, I have some Douglas firs, I have some ferns, I have some weeds, I have some grass, I have some squirrels, I have some raccoons. And so I would say I have a community there.
What's the difference between the community and the ecosystem?
So then if I add in the rain and the sunshine, the abiotic or non-living components,
Okay.
then I would say an ecosystem. So when we look at this, a photosynthesis converts carbon dioxide in the air to sugar. So this is kind of, we know we're not, oh, that's funny, population is twice. I didn't write.
I was just about to say that too.
I'm cracking up. Let's see what happens if I say edit the quiz, and then it'll give me that question right here. Oh no, let me see the questions, please. Oh no, that's right, it's from the bank. It's not going to work that way.
So it's neither one of them.
let me see. It may.
It's fine. We understand that there's two populations.
Yeah, I was just seeing if I could weed it out, but see, this one actually is a different set. They're not in order. So I'm going to go back. I'm going to get out of here.
The good thing is population isn't the correct answer, so we're good to go.
It isn't the correct answer, but I need to go back and I need to find that particular question so I can fix it. Oh no, I need to go to preview again. And I've tried to remember where I'm not in the same place. Questions. There we go. All right, I had to go back one more. So now we're all right. So when I read this, I see the focus is on different living organisms, and so I would choose community. We are talking about converting the air into sugar.
Yeah.
But I'm looking at all of these organisms interacting, and so the answer that I would choose here is that it's a community.
Understood.
Okay. And I'm making a note so that I'll go back to it and I can try and get out population here. Now this one, it says which level, so this is a different version, which level in the hierarchy shown is a community. So the savanna is the ecosystem. It's all the organisms that live in a particular place that has a given climate and a certain amount of rainfall and a certain amount of sunlight and is a certain distance from the equator. So this is the ecosystem, the organisms plus the abiotic components. But all the organisms in the savanna, that is the community. And a single group of cheetahs would be a population.
Or population.
This would be a
No, I-
population. It would be a population. All of the cheetahs would be a species. So
Yeah,
it would
I
sort
get
of depend on how that one was described, whether you would choose species or a population. But a group of cheetahs in one part of the savanna in Africa is just a population.
Okay.
And a given cheetah is a member of the cheetahs, that particular species of cheetah, right? Where species just explains, you know, a group of organisms that can only reproduce viable offspring with another member of that same group of organisms. Okay, number 5, it says, in presenting data that result from an experiment, a group of students show that most of their measurements fall on a straight diagonal line on their graph. So when I read that, I think, and I'm going to show you with my arrow key, if you follow it from like left to right, it's just going straight up and down. But 2 of their data points are outliers. So that would mean that the line doesn't go near them. So they're like up here and down here. They're just odd. They don't meet or match the pattern. So what do? So this is just getting at when you're applying the scientific method, do you get to selectively check your data? Or do you have to use, or do you have to use all of it,
You have to use
right?
everything.
Right. So there are scientists who are like, oh, let's just get rid of that. Let's apply this sort of statistical analysis that will remove it. And I can tell you from personal experience with my ecology experiment studying fish underwater, that when the fish hit under the platforms, that didn't mean I could just throw out those particular platforms. I had to throw out all the data that I had because I had like a confounding variable. I had something messing with my experiment and I couldn't actually see the results. And I couldn't just throw out the ones that were messed up out of, you know, like the 20 or so data units I had. I had to throw it all out and start over because I couldn't analyze any of it with any sort of validity. And all I could say is these fish I was studying were really good at hiding. That was the only outcome from that. So it says, like this one, show all results obtained and then try to explore the reasons for the outliers. At first glance, that's totally where I'm like, yes, that totally makes sense. You have to use it all. And then what I do is I read through everything else and make sure that I didn't miss anything.
That's just good test taking though.
Yeah. Change their experiment so that the outlier points are eliminated. No, that's screwing with your data and manipulating it. Average several trials, rule out the improbable results and do not show them in the final work. So that's, I can tell you that happens sometimes, but it shouldn't. With mine, I just had to do a data dump because of the confounding variables. And I basically, it says redesign the experiment using a different hypothesis. I didn't have a different hypothesis. I didn't do this last one. If you don't have good data, you can't reject your hypothesis and get a different one. You have to analyze the data that you have. With mine, I essentially had no data because it was fish on these habitat platforms, and I was supposed to look at predation rate, but if they were hiding, but only on some of the different platform types, then I couldn't compare them and I had no predation rate that I could effectively analyze. And so I didn't redesign my experiment. I had to redesign my units and figure out how to prevent them from hiding because there had to be equal opportunity. It was kind of fun. So I didn't see any, you know, change the values on the graph. Again, that's manipulating, you know, your data. So a lot of these are like data massaging. And so I would stick with this first one. Okay, next one. Francesco Reddy doing the experiment with jars, and he has 4 that are open and 4 that are sealed. And this is about spontaneous generation. People used to think, you know, I throw rags in the corner and a few days later, voila, rats. So the rats came from the rags. Well, things weren't as sanitary a few hundred years ago, right? So it makes sense that a rat came, nested in your dirty rags in the corner, and a few days later, what did you have? Baby rats, right? So here he's looking at maggots from flies, and he's basically saying you don't get something from nothing. So he's got 4 sealed jars, and then 4 that were open, and then he repeated it, and he took 4 jars and he covered them with gauze so the flies couldn't get to the meat. And so basically, what is the independent variable? What is the thing that is different about his groups of jars?
The jar is sealed with the gauze.
Yeah, the type of cover, sealed or gauze or like completely, you know, open, right? And so I would choose this one, the type of cover. This is what's different about them, okay?
Yeah, I got that one wrong.
Okay. So independent means the thing that you decide beforehand that makes your different treatments different. And this is my youngest. Hi. Do you need to go watch cartoons?
No.
Of course not. Okay, can you go play, please? I'm going to pause. All right, here we go. So this is one where it gets tricky because you have to remember what independent variable means, and then you have to apply it to the scenario, okay?
Yeah.
And then dependent variable, that's the thing that he is measuring. He monitored the jars to record whether or not maggots appeared in the meat. So the appearance or not of maggots, that would be the dependent variable. And things that he probably controlled, well, we see 8 identical jars, so the jars were all the same. That's one of the things we'd call a control variable. Okay, so this next one, the NIH set up a study looking at vitamin C and time to recover from a cold. And the question says, what is the experimental group? So we have a group A and a group B. And when we say experimental, we would think this is the group that is getting the treatment as opposed to a placebo. So group A gets 3,000 milligrams of vitamin C, group B gets 3,000 milligrams of placebo. So which of them would you call the experimental group?
Group A.
Group A, okay, good. And then if I wanted to say what the independent variable was in this one, it would be the presence or absence of the vitamin C. Yeah, and then the dependent variable, the thing that was getting measured,
was the length of time the cold.
Yeah, the length of time the cold lasted. Excellent. Okay. All right. So we already covered independent variable. We can scroll past that one. Dr. X is interested in the effect of weight training on the density of mitochondria and the skeletal muscle of athletes. So she has a big sample group, 100 18-year-old male swimmers as her test population. Oh, that's interesting because that's just swimmers and just males. And she's got she's got mitochondrial density from the calf muscles. So basically pulling, you know, a sample, a tissue sample, and looking at how many mitochondria there are in cells before and after weight training. So the independent variable is basically what's the difference here? We've got a before and after with a given group, so there's not actually an experimental group and a control group. We have a group that's the same group is being used, so it's a before and after. So what's the difference between the before and the after? That's your independent variable.
Oh, yeah?
Yeah.
Be the number of mitochondria, correct?
No, the independent variable, the difference between the before and after is the weight training. What is the thing that we think is going to make a difference? We think weight training makes a difference. So it's the thing that we're changing. There's a before weight training and an after weight training. And the thing that we're measuring or recording, the dependent variable, is the mitochondrial density. So this right here is the independent variable, and this is the dependent variable. And there's actually no true control group because it's the same group that we're doing a before and after. If we wanted to have an actual control group, we'd have to have another group of 18-year-old male swimmers and have them on the same diet and not do weight training, but do everything else and see if there was a difference. You know, measure them before and after at the same time period, and then they would be our control group. Okay. So number 10, we're jumping into chemistry. When are atoms most likely to react? And so we're talking about when will an atom participate in a chemical reaction? And that's going to be when its valence shell is not filled. So which one matches that?
When the valence energy levels are not filled?
Yeah, this one. So if you think about, if you're looking at this and you're like, oh, if I look at the periodic table and I know, why are these called the noble gases? And it's because they're on the far right column. Their valence shells are full. And so they don't need any electrons to be balanced. They're balanced as they are. And so they're inert or noble or non-reactive. And it's all the other atoms of elements where their valence shells are not full that are unstable and therefore likely to participate in a chemical reaction. Okay, number 11. What kind of bond would you expect lithium and iodine to form? So for this one, there was a Zoom recording that I have where I go over part of one of the learning assignments with electronegativity, and I believe I have a recording in there with the periodic table looking at electronegativity. So when you look at, there's a giveaway here though, Li plus and I minus, one's plus and one is minus. So what is that giveaway right there?
Polar.
Actually, when I see the plus and the minus, it tells me ionic bond, because this one is already, it's already being shown as an ion where it's given up one of its electrons, and that's why it's got a positive sign, because it has one more proton than it has electrons. And this one already has one more electron than it has protons, and that's why it's I minus. So a hydrogen bond, that's between like different hydrogen bonds are like temporary interactions between water molecules or like water and other polar molecules. And then if what you would go back to if you were like looking at a question like this and you weren't sure is you would look to see on that periodic table of electronegativity, what were their electronegativity values, and look at the learning assignment that you did that dealt with electronegativity and bonds. But I'm just telling you right now, this Li plus and I minus is a dead giveaway that it's going to be an ionic bond.
Well, it said what kind of bond would two ions make? That would be ionic.
Well, right, and it says ion and ion, so ionic bond, right? That question is actually not particularly savvy when I look
like
at it deeply.
It's almost a gimme question.
Yeah, exactly. All right. So this one says select all that apply. A molecule of vitamin B6 has the formula C8H11 and then O3. From this you can determine, okay, so this is basically looking at the thing about the learning assignment where you learned which ones had nitrogen in them. So you know carbohydrates do not have any nitrogen in them. So it says from this, you can't determine that it's a carbohydrate. In fact, you know that it's not. Would you be able to determine whether or not it has ionic or covalent bonds?
Covalent, maybe.
You can definitely tell that the number of the atoms, right? So this one, yes, 8 carbon, 11 hydrogen, 1 nitrogen, and 3 oxygen, okay? It's a vitamin, so it's actually not gonna be a carbohydrate, a lipid, or anything else like that. When we look at C8 and H11 and we look at C and H and oxygen, and you look at them on the electronegativity table, you could actually tell, you'll be able to tell that it has covalent bonds. Given the way that it's written, these carbons and these hydrogens should all be covalently bonded to each other. And the way the nitrogen and the oxygen, it's going to be an N bonded with, probably double bonded with one oxygen and single bonded with the other one, and then bonded to carbon. But you can't tell that it has ionic bonds. And I'm not trying to get you to go too deep into chemistry here. It's just, oh, hey, it's carbon and hydrogen and oxygen, and I know those are covalently bonded together. And so those are the two that you would check. Okay, next one. What's the big difference between covalent and ionic bonding? So this is where, before you even look at it, what do you know about covalent bonds? The valence electrons are shared, and ionic bonds, the valence electrons are transferred, and then there's an ionic attraction between the oppositely charged ions. So which one matches that?
First one.
The first one, yes. And then you would just kind of quickly check the other three and make sure that none of them are somehow a better match than the first one. So when I read a question like that and I'm like, oh, I know what these are, and I would just really quickly think in my head, can I answer that question before I even look at the options? And therefore I don't get confused. And then I can knock out ones that don't match that at all. But then if something like flags or catches my attention where I realize I might be off a little bit, then I check through all of the answers just to make sure. So it's just kind of some test taking strategy. Okay, 14. A covalent bond is likely to be polar when? What makes something polar versus non-polar?
The electromagnetivity.
The electronegativity difference has to be high enough so that one atom is pulling on the other atom's valence electrons unequally, right? Mm-hmm. So which one would cause that unequal sharing? If you look at number...
atoms sharing an electron more electronegative than the other atom.
Exactly, okay, great. Okay, the next one, carbon-14 is radioactive. It has 6 protons and 8 neutrons. Does anybody remember what the atomic number of carbon is?
I think it's 6.
It is 6 because it has 6 protons, right? And most carbon is carbon-12, but some of it is carbon-14, meaning it has 8 neutrons instead of 6. So carbon-14 is an isotope of carbon, meaning it's an alternate form, but it's also carbon because every atom that has 6 protons is a carbon atom. And so this is talking about one of the 8 neutrons becomes a proton and an electron is formed in the process. So how many protons would I have if a new one is formed? 6 plus 1 is 7. So after the decay, the atom cannot be carbon because the thing that determines whether something is carbon is it has to have 6 protons. So as soon as it decays and it has a 7th proton, it is not carbon, it actually becomes nitrogen, okay? So, and it's telling you it changed, so you wouldn't choose this one, that it's unchanged. And the mass, and that's funny, the atomic mass number, we don't even really get into that. So there's some additional information that's like chemistry that's sort of being thrown at you. But you just have to recognize this simple fact that as soon as you have a different number of protons, you are not the same element anymore. Okay, 16. Atoms always react in a manner that, oh, that's hilarious, mystifies scientists. Okay, why do atoms react? What was I talking about before with the noble gases? They
results in a full valence energy shell.
Yeah, they react because they're, if I'm personifying them, they're seeking stability. And so they're going to react until the point where they have full valence energy shells, whether that is shared, so covalent bonds, or whether that is transfer and with ionic bonds. Okay, great. 17. Oh,
I'm sorry, can you pause real quick? Which was the answer for that? I didn't catch it. It was the
this
third
one.
one?
The third one, yeah.
Okay.
And you can always go back and rewatch this recording and fast forward through anything that is boring or that you don't need, right? Okay, so 17, the correct bond type to the description. So before you even look, just think, hydrogen atoms of a water molecule are bonded to the oxygen atoms by these bonds. So we're talking about one water molecule. And so hydrogen to oxygen, what type of bond would it be?
Covalent.
Covalent.
It's covalent, and if it had popped up non-polar or polar covalent, you would choose polar covalent. And now this is neighboring water molecules are held together by what type of bond?
Hydrogen.
Hydrogen.
Hydrogen. Yep. Okay, you got it.
Jinx, yummy soda.
All right, number 18, uranium, a metallic element. Most of it is 238, so it has an atomic mass of 238, and some of it is 235. So what would be the difference between 235 and 238? What gives something a different? The mass is different. So atomic mass is protons plus neutrons. And to be uranium, what has to be the same about these two atoms? They have to
Different
have
number of neutrons.
What has to what I started out was what has to be the same to kind of get to the answer.
Number of protons.
Yeah, to be uranium, the number of protons has to be the same. So X number of protons, whatever it is that uranium has, plus X number of neutrons gives the atomic mass. And for 235, it's the set number of protons plus the certain number of neutrons. And for 238, it's 3 more neutrons. So the difference is that 235 has 3 fewer neutrons than 238. I guess Paw Patrol wasn't scintillating enough. She's back. Okay.
We'll do that.
Yep. I know what she's after. She wants Peppa Pig on my phone. You're gonna get away with a lot of stuff right now. Go sit on the couch, please. Thank you. All right, number 19, looking at leucine, an amino acid, and it says, what is the arrow pointing to? So what does a solid single line represent when we're looking at chemical structure?
Single covalent bond.
A covalent bond, and is it polar or non-polar? Does it matter?
That's what I'm confused by.
Okay. So we know here, carbon to nitrogen, that's going to be non-polar covalent. When we're talking about nitrogen and hydrogen, nitrogen acts like oxygen. It is very, very electronegative.
Okay.
So this is going to be a covalent bond, but it is going to happen to be a polar covalent bond. So this question
So there's no actual information as far as the lines go as to if it's polar or non-polar. You just need to know that ahead of time.
Well, this was getting at 2 things here. Do you know that those solid lines represent covalent bonds, and that a single line is a single bond and a double line is a double bond?
I know that.
And then the other piece that you have to catch here, and you're like, oh, there's more to it, is you see this, and then you see this. And so you're like, oh, okay, it's between nitrogen and hydrogen. If I look at electronegativity, if I look at the periodic table, nitrogen and oxygen are the most electronegative elements that are found in biological organisms. And so when I see nitrogen and it's not bonded to a carbon and it's interacting with a hydrogen, if it's oxygen or if it's nitrogen interacting with hydrogen, then this is going to be a polar covalent bond.
Okay.
Good?
Yes.
All right, so this is octane because we got 8 carbons, and what are all of these bonds between the carbon and the hydrogen?
Single covalent.
They're single covalent, but look,
Non-polar.
yeah, they're only non-polar covalent bonds.
Is that because of the carbon?
And that's because carbon isn't really electronegative and hydrogen prefers to hang on to a single electron. And so the carbon is sharing equally with the other carbon and it's sharing equally with the hydrogens on either side. And so they're non-polar covalent bonds. If you were to do the electronegativity calculation and look at the difference between carbon and hydrogen, it's different, but it's not significant enough for it to make a polar covalent bond. It's going to be non-polar. And in case you're wondering what that is, I'm going to pop up here. I'm going to have Google be my friend. And on my other screen. Does that web page show up?
Yes.
Yes.
Yes, okay. So if I type in electronegativity periodic table, I can grab something. I was hoping it would just open it for me. Oh, it probably opened it above, sorry. I'm waiting for it to open up the image for me. Amongst all the ads, I get all the ads first, of course.
To the image search, it'd be easier.
All right, and just do this.
Yep.
Oh, it's not gonna let me make it big. I don't know if it's gonna let me make it big right now, if I'm gonna be able to figure that out here. If I go, is that a little better? Can you see how it's really red over here? Oxygen, fluorine, and chlorine are the most electronegative elements, okay? Those, they're pretty high up in the rows of the periodic table, so they don't have a whole lot of valence shells. So their valence electrons are actually fairly close to the nucleus, so they're held pretty strongly. And they have a high number of electrons, because they're all the way over here on the right, far right columns of the periodic table. And so these three have the most electronegativity, and then they're followed, you know, close behind by nitrogen and bromine and krypton, actually, which is kind of funny, even though it's a noble gas. But of all of these that are really electronegative, what do we find in living organisms? Oxygen, nitrogen, and carbon. But carbon is just 2.55. So when we compare 2.55 to 2.2 of hydrogen, the difference is only 0.35. So it's less than 1. So it's not enough to make a polar covalent bond. And so this is something that you can pull up and look at, and it goes in conjunction. I want to say it's learning assignment number 3, but I'm not absolutely positive on that. The one that had the electronegativity table calculations that you had to do. Okay. All right, so we ready for 21?
Yep.
Okay, I'm just looking at time here, trying to make sure we get through as many as possible. A strong acid like HCl, hydrochloric acid. So before you even think about that, what does a strong acid do with its hydrogen ions? Does it keep them bound tightly, or does it release them into the solution?
Keeps them bound tightly.
It releases them.
It was 50-50. I was making one shot or the other.
And
you're bold for trying.
Yes. See, there's no harm in being wrong here. You lose nothing, and in fact, you gain because you learned something.
Exactly.
Right? Too bad you don't get $50 with every wrong answer too. But hey,
Oh yeah.
one can dream. What I know, I'd be rich too. One can dream. Okay, so a weak acid doesn't release many hydrogen ions into the solution. And a strong acid releases more, and the strongest acids release more hydrogens than intermediate strength acids. And if I were to compare HCl to like something that had like 4 hydrogens bonded to a single different atom, that would be an even stronger acid than hydrochloric acid. Because if it's going to dissociate completely from its hydrogens and release them all in solution, the number of hydrogen ions in a solution determines how acidic that solution is. And so when I think about that, a strong acid does what with its hydrogen ions? It releases them. So which of these seems to apply most completely to that? And I would look at, I think, this one right here dissociates completely in an aqueous solution. So aqueous meaning in water. So that makes sense. Reacts with strong bases to create a buffered solution. That may be true because when I combine equal amounts of hydrochloric acid and sodium hydroxide that are the exact same concentration, I'll have a neutral solution. But I don't know about a buffered solution. So you're like, ooh, that's playing with what you know about buffers a little bit there, and maybe too deep. So number 2 looks good. Let's look at number 3. Dissociates completely in aqueous solutions and is a strong buffer at low pH. Weak acids make better buffers because they will let go of their hydrogen ions and grab them back again, where a strong acid is just going to let them go. So I still like number 2. Number 4 here, increases the pH when added to an aqueous solution. What does hydrogen ion do to the pH? Does it make the pH go up or the pH go down?
Down.
It goes down, so it's not increases, it's that it lowers. And this is a strong buffer at low pH. A strong acid is going to have a low pH anyway, so why are we even talking about strong buffer? So the thing that's tricky here that makes you want to choose this is because this says strong and this says strong. Don't be fooled, okay? A strong acid means, quite simply, that all of its hydrogen ions separate in water. And so we'll stick with 2 right here. 22 is getting at, do you know on the pH scale what happens when you jump from one number to the next? So if I go from 5 to 6, where I'm becoming more basic, my hydrogen ion concentration goes down by a factor of 10. If I decrease my pH, then my hydrogen ion concentration goes up by a factor of 10. So when I look at pH 3 and a solution of pH 1, so is pH 1 more basic or more acidic than pH 3?
More acidic.
More acidic.
It's more acidic because the number is lower, it's further to the left. And how many steps to the left? Well, I go from 3 to 2 to 1. So it's 1 step from 3 to 2 and a second step from 2 to 1. And that's not 20 times more acidic. It is 10 times 10. So it's 100 times more acidic. That was on one of the labs, the second lab that you did. There was a table and an explanation about that, and also some information on one of the learning assignment that dealt with acids and bases. Okay, in this one, 23, this equation looking at hydrogen and oxygen making water, it says the H2 molecules are blank and the H2O are blank. So on the left side, with the arrow proceeding to the right, this left side is always the reactants, and this right side is always the products. So the H2 is reactants, and the H2O is the products. Product or products, it's okay that it has the S because there's 2 of them. All right, 24, partial negative charge in a molecule of water occurs because, so before you look at it, what does it mean to have a partial negative charge? Do you remember the funny symbol with the slight positive and the slight negative, the Greek symbol?
I don't know what symbol you're talking about, but I
It looks like
understand it. I just don't know how to explain it.
it looks like a figure 8 almost, but it's not closed. Here,
So yeah, I understand what you're saying, what you're describing.
okay.
Be like a with a U on it, kind of.
Yeah, it's almost like a funny G, or we'll do this again. Water molecule.
I'm a visual learner, so I have to see it.
All right. Whoops, that was me. Do you see this second one right here?
Yep.
Ah,
It's this right here. So here's the partial charge. This is showing that hydrogen is partially positive and oxygen is partially negative. So that's right as soon as that question says that, this is what you have to visualize. And so this stick right here, this line means that it's a covalent bond, but because it's partially negative and partially positive, that's telling you that this is a polar covalent bond. And why is that happening? I've got a video that you hopefully watched that explains that the electrons around oxygen and around hydrogen spend way more time orbiting oxygen than they do either hydrogen. And so that creates an imbalance of electron distribution. Overall, the charge is still neutral, same number of protons as electrons, but the charge of electrons running around oxygen more of the time makes this region of the molecule slightly negative and these two regions slightly positive. And so the electrons are being shared, so it's a covalent bond, but because their distribution around the various atoms in the molecule is unequal, then it's a polar covalent bond. So if we go back to that question with that image in mind, the partial negative charge in a molecule of water. Now, where was that negative charge? Was it on the oxygen or the hydrogen?
Oxygen.
it was on the oxygen.
It was on the oxygen, and it occurs because, now what matches what we just discussed?
The electrons shared between the oxygen and hydrogen atoms spend more time around the oxygen atom nucleus than around the hydrogen atom nucleus.
Yes. Now, what would help you answer this quickly? A, you've got your notes right there and you can flip to the diagram that you already have, or B, you have some note paper, and what do you do really quickly? You're like,
notes.
oh, it's a molecule of water, and you either refer to your notes or you sketch a molecule of water and the charges really quickly so that you can use that diagram to help you answer the question like we just did. Does that help in a little with test taking strategy and how to utilize your time and make sure that you access the part of your brain that you've put on paper or wherever you've saved it electronically? There's no shame in using, when it's available to you, the content that you've prepared and continuing to layer that information in a test. I've taken plenty of tests where that's not an option, but when it is, there's nothing wrong with using it. Okay, 25. The bonds within a molecule, so inside a water molecule, are those hydrogen bonds?
Nope.
No.
Nope, they're false. Those are polar covalent bonds. Okay, so what happens when ice melts? Why does ice melt?
Water molecules become less tightly packed.
They become
Well, I guess-
less tightly packed, absolutely, but what happens? When water is ice, each water molecule has the maximum 4 hydrogen bonds formed. When I
Hydrogen
add
bonds are broken.
yeah, when I add energy to the water, so I put some ice cubes out on the hood of my car, right? Then the sun and the heat from the hood of my car are introducing energy that is destabilizing those hydrogen bonds and causing them to break. And as soon as they break, the water molecules are allowed to move. They're no longer solid. And so they can move, they're less tightly packed. And so, let's look at this. What would I have to choose here? And this question, I would say, should be rewritten to show that as ice melts, hydrogen bonds are broken. And do the water molecules become less tightly packed, or do they become more tightly packed?
They become less, correct? Because wouldn't the bonds be weakening?
Look at less tightly packed. When is less tightly packed is another way to say less dense. When is water the least, have the least density? When does water float?
When it's oxygen or when it's a gas.
When it's
Ice.
when it's ice. Solid floats. The maximum number of hydrogen bonds happen at 0 degrees Celsius. Believe it or not, water is actually the most dense at 4 degrees Celsius. When it's cold but not frozen, it actually sinks, and it's really important for circulation in lakes and the oceans and bringing oxygen to the surface and then down deep below. But that's because I'm a marine ecologist. That's why I think that's cool. But 3 and 4 here are actually saying the exact same thing. Less tightly packed is the same as less dense. That means pushed further apart. When ice melts, what do the water molecules do? Do they stay far apart, or do they melt and become a fluid and become closer together? More dense.
Closer together.
Yeah. So because this almost looks like a double negative here, and so it's a little bit tricky. But both of these are what happened when you get ice, which is not what is happening when ice is melting and you're getting a liquid. So its hydrogen bonds are broken.
I have a question on that.
Yeah.
So when it's asking about water molecules becoming less tight, and the second one saying the water becomes less dense, so is it talking about the water as a whole versus like the actual molecules? Is that the difference in the answers?
It's just.
They're saying the water becomes less dense. That's making me just kind of like in a general sense the ice is melting, but the water molecules becoming less tightly packed is more of like a microscopic.
But when ice melts, the water becomes more dense. The water molecules become more tightly packed. So neither one of these applies to the 3 words given. Because as ice melts, water molecules become more tightly packed. And in essence, the water becomes more dense because liquid water is more dense than solid water, which is ice. If I were to rewrite this and address it, I would say the liquid water becomes less dense, but it's still false as written. Just not as technically correct, but sometimes the false answer is not meant to be technically correct, it's just meant to be false. And that is just false. Okay, looking at this here, if these are water molecules around the outside, what would all of these big dark blobs be?
Negatively charged.
They would be oxygen, right? And they would be what kind of charge?
A negative charge.
They would be negative, so this thing in the middle is?
Positive.
Positive. There you go.
Could have sworn that's what I put, but I remember getting that one wrong. I don't know.
I'll check it. It's possible that one got miskeyed and nobody brought it to my attention last term, so I didn't get to make a correction or a deletion with the curve. So I'm just writing Q27. Okay, 28. Looking at this one, it's kind of similar, but an alternate version, right? So now we have all the little positive hydrogen atoms, not ions, facing to this. And it says the large molecule is most likely. And so then you have to look and you're like, oh shoot, can I remember? What is cation? What is anion? And so if you can't remember, then what is your friend? Google, and you really quickly go, oh crud, I can't remember, and you pull it up and you go, what is an anion?
Hey Cortana, what's a cation?
And it says it's a negatively charged ion. It's negative. Okay, so if an anion is negative, cation is positive. So what is this guy here?
Negative.
It's a negative, and so what fits?
Yep.
An anion, right? And so see how you still answered that in like the minute and a half you had, because you're like, oh, I know it's one of these, but which one was it again? So it's either in your notes or Google is your friend. See how that works?
Yes.
Okay, good. That's a dumb question to ask, but I guess there's always, I hope you would tell me if you didn't get something. Okay, 29. The temperature of evaporation is much higher for water than alcohol. So what does that mean for the temperature of evaporation for water being much higher? When
It takes. What was the question?
You were about to answer it, so I'm saying which one would actually evaporate at a lower temperature, the water or the alcohol?
Alcohol.
The alcohol.
The alcohol. Good. Okay. So without knowing more about the chemistry of alcohol, which of the following is the most logical chemical explanation for this phenomenon? So, you're getting at like, what must be different about water than about alcohol? Well, you know, water has hydrogen bonds between those different molecules. So maybe that's the difference. But let's look here. Ionic bonds form between alcohol molecules. These are the weakest type of bonds and are easier to break than the hydrogen bonds. Actually, if I were to rank them, covalent bonds are the strongest, then ionic bonds, and then hydrogen bonds, which are external bonds, are the weakest. So that one doesn't quite fit. Okay, alcohol has a higher surface tension. Well, what does surface
I
tension
already know
mean?
that's wrong.
Yeah, you know that's wrong because surface tension is because of hydrogen bonds between water molecules, so alcohol doesn't have any surface tension. All right, fewer hydrogen bonds form between alcohol molecules. Now, alcohol does have OH groups, so it does have polar ends, so it does have the possibility to maybe form some hydrogen bonds, maybe, so that makes sense. But if it has less than water, would it need less heat to make it evaporate and separate and break those hydrogen bonds?
Yes.
Yes.
Yeah, so this one is making sense. Alcohol molecules are more cohesive. Well, if they have fewer hydrogen bonds, they can't be more cohesive. So the only one that makes sense that would allow for faster evaporation is this third one here. Okay, so let's see. It's 8:15 about, and I'm thinking this is about pH. Then how many more do we have that are up here? There's only 41. So what question were we on? Should we just do the last few really quickly?
Yeah.
Knock
Okay.
them out.
All right. So 30 here. And notice we've only got like a sample of like all of the questions, right? We don't have absolutely every question up here. Okay, so let's go to, we were at 30, right?
Yep.
Right.
Okay, so after reading the paragraph, answer the questions. Acid indigestion, quick fix.
I actually got this one wrong.
Excess stomach acid. So your pH in your stomach is really low. And if you have lots of stomach acid, then the pH of your stomach is probably even lower than normal low. And if you take Pepcid AC or Alka-Seltzer, then those are probably going to be what? Somewhat basic to neutralize some of the acid in your stomach. So you hypothesize that a tablet of Pepcid AC can neutralize more stomach acid than a tablet of Alka-Seltzer. So it's basically getting at which is going to do a better job of neutralizing your stomach acid, the Pepcid AC or the Alka-Seltzer.
Yeah.
So rather than pulling your stomach out of your body to test it, right, or using yourself as a guinea pig, it says if you place a tablet of each antacid in a beaker of acid and initial pH of 2, what would support your hypothesis? So what you need, if Pepcid AC does a better job of neutralizing, then it will pull the pH higher than the Alka-Seltzer.
Makes sense.
Because a higher pH is closer to 7, closer to neutralize. And
Yeah.
for a stomach, it just means make it slightly less acidic. So after 1 hour, Pepcid AC is 9.2 and Alka-Seltzer is 8.3. So this one went more basic than this one, okay? And this one, the next one, it says they're equal, so that wouldn't support it. And after 1 hour, 7.9 versus 9.6, so this is kind of the reverse. And then this one, it took 3 minutes for the one tablet to dissolve and 5 minutes for the other one to dissolve. What is the
That's not what I was
dissolving?
asking, though.
Dissolving like would tell you how fast it acts, but you're right, it's not what it's asking. So
Yeah.
this is the one I would choose.
That's why I'm wondering how I
Because-
got it wrong, because that seems
Yeah.
pretty obvious.
It does seem pretty obvious unless you're, you know,
In a rush.
unless you rushed or you accidentally clicked on the wrong thing. And on a computer test, when you incorrectly click and you don't notice, it bugs you later. So I'm just gonna double check that one to make sure too, since you brought it up. Okay, 31. Cellulose and starch, how are they different? Well, they're both glucose monomers, but if you went back and looked at the PowerPoint that I did and you went back through a couple of videos, you'd actually see that cellulose and starch have different orientations of their glucose monomers. So this one's false because they're both glucose. And it says starch is formed by plants and cellulose by animals. That's false because plants make both starch and cellulose. Starch is energy storage and cellulose is structure. And you can eat all the celery you want and never like get any calories because it's the wrong kind of glucose bonds. And it says most animals cannot break down cellulose, whereas starch is easily digested. So that is true. And then this one, cellulose is highly branched, whereas starch is unbranched. This is false because they're both greatly branched. This one doesn't even ask you about alpha versus beta glucose, although that is on one of the learning assignments. So this goes directly into what I was saying about how many of you are going to go like chop down a tree and like just munch it up. You can't. And even a cow, the only reason it can digest grass is because it has special bacteria that are able to digest or break the bonds between the glucose monomers in the cellulose. And that's the only reason cows and other ruminants can eat grass. Hold on one second. Alma, turn it down, please. Thank you. All right, 32. A protein, so more on biomolecules. A protein containing more than one polypeptide chain has what level of protein structure? So let's just walk through this. Primary is the sequence of amino acids. The order in which those amino acids are joined together in a chain we call primary structure. And it determines the final structure of the protein and how it actually functions, but it's just the order that those amino acids get bonded together. Secondary is when the way that those amino acids get bonded in patterns to form alpha helices and beta pleated sheets. So it's links between the amino acids and essentially a lot of hydrogen bonds. And tertiary structure is when we get additional hydrogen bonding and interactions between R groups of those amino acids, and we have a three-dimensional, full three-dimensional shape. But as soon as we add multiple subunits of polypeptides, then we say we have quaternary structure. And a protein can be complete when it has tertiary structure, and some proteins are only complete and functional when there are multiple subunits that are interacting together. Hemoglobin is an example of that. There are 4 subunits that are necessary to work together to be in your red blood cells to bind to oxygen. We okay with this one?
Yep.
Okay. 33. What does di mean?
2.
2. So what makes sense? Oh, wait, they all say 2. So disaccharide, do you get it from 2 starches or 2 monosaccharides?
Monosaccharides.
2 monosaccharides. So you've got 2 monosaccharides, and then when do you get it? Is it by dehydration synthesis or by hydrolysis?
Dehydration synthesis.
Yep, so we just took that apart. We're like, oh wait, they're all 2, so that's not helpful. Now I've got to do monosaccharide versus starch, and then I've got to look at dehydration synthesis versus hydrolysis. So there's several layers to that. 34, what's a phospholipid? Okay, so in here, it's a lipid, so it's one of the categories of biological molecules, and it's going to have fatty acids, but why phospho? It's going to have a phosphate group, at least one, so that eliminates this one. That one can't be true, and now you have to remember, does a phospholipid have 2 phosphate groups, 1 phosphate group, or 3?
Oh, man.
Unclear.
It's 1. So this one, there should be a phospholipid in one of the learning assignments and also on the PowerPoint that I have posted. So you can see that there's a 3-carbon molecule that's the glycerol backbone. And so there's 1 glycerol molecule and there's 1 fatty phosphate group and 2 fatty acids. A triglyceride is the normal lipid structure where it's a glycerol and 3 fatty acids. But our cell membranes are composed of phospholipids where one of the fatty acid chains is swapped out and instead we have a phosphate. And so you have a polar region, the phosphate group that can interact with water, and a non-polar region, the fatty acid tails, the long hydrocarbon chains that avoid water. And so the tails, and I know you can see my hands even though I can't, if the palms of my hands are the phosphate groups and these are the fatty acid tails, the fatty acid tails will orient toward each other in the interior and the phosphate groups form the outside. And picture that as a sphere where this is happening all the way around and you end up with a double-walled barrier that creates a wall of a cell, or the membrane of a cell is more accurate. So it's this one, 34. All right, what makes a fat hydrophobic? What makes it not interact with water?
Is it the carboxyl groups?
Not quite.
The non-polar hydrocarbon chains.
Yes, they're
That's what I said.
non-polar, so they can't form polar. There's no polarity, so they can't form hydrogen bonds with water, so they won't interact with water, and we call that hydrophobic or water-fearing. Okay, 36, pepsin is a protein that breaks down polypeptides in the stomach. And so we're thinking about, all right, it's in the stomach. It functions at body temperature at a pH of 1.7, which is pretty dang acidic, and a salinity of 0.9%. What would denature pepsin? What would make a protein not work? If I change the temperature, if I change the pH, or maybe if I change the salinity? So what would denature it, make it not work?
Wouldn't a basic solution cancel out the acidic solution?
A
Or
basic
cancel out its
solution.
acidity?
Yeah, if I put it in a different pH, it might not function well and it could fall apart. If I put it in something that is really salty, it might change it. Oh, body temperature is 20 degrees Fahrenheit body temperature.
No.
Dead
You're
body
gonna be,
temperature?
not your body temperature. So if I freeze it out, it probably won't work. What about a temperature of 110? What happens
That's
to your
feverish.
yeah, when that's like dead fever.
That's dead fever. That 106 is almost dead fever. So
If you have a temperature like that, part of the reason you're gonna die is because your proteins stop functioning. Your proteins pretty much do all the work of your cells. And so if your proteins can't work, bye-bye cells.
would that also cause rhabdo?
Cause what? Sorry.
Rhabdomyolysis? Would that cause that? Increase in body temperature?
I don't know what that is.
It's the breakdown of muscle tissue from like excessive exercise. I treated one dude when I was in the military with it. He like completely fainted. Like I thought the dude was gonna die on me, and turned out he had rhabdo. It was like death of like proteins in the muscle tissue.
I wonder if that's from excessive exercise and like lactic acid buildup. I don't know.
That would do it.
I don't know. I can't answer that competently. Sorry.
No worries.
By the way, pH of 1.7. So this is already really acidic. So we're not going to choose acidic pH. But if I neutralize it, that would do the job too. So I
Okay.
choose all of these, but that one. All right, 37. We're getting really close. Okay, how do 2 amino acids get attached together? Well, we know it's a dehydration synthesis, and here's a peptide bond. So what do I call this? We know it's dehydration synthesis and a peptide bond, but that's not my choices here. So what is this thing called? The COOH? It's a carboxyl acid group, and this NH2 is an amino group. So what 2 things are being bonded together in a peptide bond?
An amino group and a carboxyl acid group.
Yeah, and it's stated flipped in reverse, but it's still true because it's still 1 and 1.
Is there like an easier way to remember that? For like how they connect? Or do you have like a little like cheat sheet that you used to remember?
There, for like, well, I know that a peptide bond is the special name for the covalent bond between two amino acids. But if you were to download them, I just have them here on my laptop. Oh, come on over. I gotta remember which one it is. It should be this. So you have all of my PowerPoints in the Canvas modules, and you can download them. And this one, if it opens, there was a cheat sheet in here where they're named. I'm just waiting for it to populate, and it's being kind of slow. Maybe I have too many things open. So slide number 8 here shows you all the names of the functional groups. So that might be a helpful place to go back to. So as far as a cheat sheet goes, that's sort of it. It shows a phosphate and an amino and a carboxyl. Okay, and then this one right here, slide 31, shows the carboxylic acid and the amino group and the bonding together. So if you had, you know, the PowerPoints open, this is when having another screen is really helpful when you're taking a test, and you just kind of pre-open some of the things like this and have them organized so that if you need to refer to something, you can do it really quickly.
I will second that statement. Because I use 2 screens, and it's like one of the best investments I've ever made.
Yeah, it is pretty nice. And I used to work just on my laptop, and my eyes don't care for that much anymore. I still don't need glasses, but someday soon, probably. All right, so sucrose, C12H22O11, and it's talking about linking glucose to fructose, which are true. Okay, sucrose, glucose, and fructose, would you agree that they're all carbohydrates? They all have the CHO 1:2:1 ratio, so I would choose that. What would you call it when you bond sucrose and fructose together? Sorry, not sucrose. Glucose and fructose. When you bond glucose and fructose together, would you call it dehydration?
Yes.
Yes. Okay, sucrose, is it a monosaccharide? Is it just one or is it 2? Because I'm putting glucose and fructose together, so it is a disaccharide. Yeah. Okay.
The disaccharide is 2 monosaccharides.
And then
That's
check this out. Glucose is C6H12O6 and fructose is C6H12O6. So guess what they are?
The same thing?
Yeah, isotopes, ooh, they're not isotopes, they are
Polymers?
they're isomers. That's actually kind of rude.
Oh, that's a mean one.
Yeah, because you're like, oh yes, but then actually if you look at this here, they're isomers rather than isotopes. Yeah, that's rude. Okay, sucrose, glucose, and fructose are all polymers. No, because glucose and fructose are monomers. Okay, is glucose a monosaccharide?
Yes.
Okay, so just those 3. And if for some reason the test marks this, if you get this and the test marks it wrong, somebody tell me and I'll like give everybody 2 points, regardless of whether they had this question or not, because I'm not sure. I'll go look for it in the key and make sure nobody miskeyed that one because of that stinky isotopes versus isomers. All right, so 39, yeah. If you have a vegan diet, what would you never eat?
Cellulose?
No, this is plants. You would eat celery, you would eat potatoes. You would actually, cholesterol is naturally made by many organisms. Oh, that's interesting. because this is a, and phospholipids, I absolutely think that this is a bad question. I think somebody, the person who wrote this, and I didn't catch it, I think that this should, if you were a vegan, you would never have protein, but there's no protein here. And actually, if you wouldn't eat animal protein, but you would be eating cell membranes, so you would still have phospholipids. So that I find a really aggravating question, and I'm inclined, because it's in there, to just tell everybody that your test just got your whatever your test results are. There's an automatic bonus 2 points on your test to your even the last when you take it the third time. And I'm going to go find that question and eliminate it. I don't know which question bank it's buried in, but I'm going to find it.
So one of those answers should technically be correct then?
Well, it says, what molecule would you never eat? And that,
Whoever put it on there had to think one of those was correct. So I wonder which one.
yeah, well, I'm thinking they think cholesterol is correct, but there's cholesterol in plants. So whoever it is, that's what they were thinking, but it's false. Okay, so this one, big biology exam, a snack, you buy snack cakes, huh? And then, what molecule do you expect to be the most abundant in your digestive system?
I want to say triglycerides, but I don't want to be wrong.
Well, 4.5 grams of fat versus 27 grams of carbohydrate. So this is like 6 times more carbohydrate. So I would actually expect there to be lots of simple sugars in your digestive system after you ate this. Because there's no fiber, so no cellulose, and triglycerides are from fats, so those would be their amino acids. You wouldn't expect there to be many amino acids because there's 1 gram of protein, but most abundant would be the simple sugars and monosaccharides. All right, last one here. So if your pH of your stomach, what do you think would happen to the protein that you ingested?
Oh man, it's either 1 or 4.
Yeah.
I would say it becomes denatured. But the only other thing I can think gets broken down is because of it would depend on the protein, I guess.
So this is the one that I would look at first about it. When we're talking about it being extremely acidic, it would become denatured, it would be unfolded, and then eventually it's going to get broken down through dehydration reactions.
So which one is it?
Take the first one, that it's denatured.
Okay.
So not the one that my mouse is currently clicked on where I said I wouldn't do that one.
Okay.
Because that follows. Okay. All right. So